So, dear learners, take talks education. Let us proceed with biology. Of course, we are still moving on with the codominance. The other time we did a course involving codominance, and here is an alternative method of carrying out the same course, whereby we shall not use a letter to represent color. I understand scholars, you normally meet those two variations. So, when you meet those two variations, then don't get surprised, don't get puzzled. Of course, both of the two are accepted. Sometimes you can choose a letter like C to represent color, like in the previous episode or video, and then you can decide to do it or to do the cross without color. So now, let us see what happens here. So we shall write that let capital R. Of course, the question reads, when a snapdragon antherinum with red flowers is crossed with another snapdragon with white flowers. Of course, all plants in F1 generation bear pink flowers. Show the results of the F1 and F2 generations. We can as well say, explain the results of the cross. So now, solution. Solution, we say, let the allele for red flowers, B, allele for red flowers, B capital R, and we say, let the allele for white flowers, B capital W. You can see that this time I have not used capital C to represent the color. So now, parents, that is now F1 generation. F1 generation. F1 generation. F1 generation. So we are moving on. Possibly I can make it here. F1 generation. F1 generation. Say parents phenotypes. We have pink flowers. We have red flowers. We are crossing them with red flowers. Red flowers being crossed with white, not red. Being crossed with white flowers. Then parents genotypes. Of course, the parents, are diploid. We have RR, the gene for red flowers. We are crossing it with the gene for white flowers, which are capital W, are shown on the board. Hope you are seeing what is happening. Then, during meiosis, the gene segregates to form R, also form R. These are carried in single gametes, which are haploid. So, in a similar way, this gene also segregates or separates to form allele W, and allele W are shown on the board. Then next will be crossing. The genes now can combine or can fuse during fertilization, which is random, whereby we shall make cross lines as shown on the board. So, at fertilization, then this is what happens. We make our cross lines perfectly as shown on the board. So, after making that, I've said this is during random fertilization. Random fertilization. Random fertilization. So here we have F1 genotypes. F1 genotypes, that is 2N. Then here, this all offspring will have same genotype, RW, like that. R, capital W, capital R, W, R, W, and R, W. So, that is the offspring phenotype. Then we say F1 phenotypes. F1 phenotypes. Then we say all pink flowered. All pink flowered. All pink flowered plants. On going out to F2 generation. F2 generation. F2 generation, we have parents phenotype, pink flowers, we are crossing them with pink flowers. So, this is what is happening on the board. We are now carrying out what we call the F2 generation. F2 generation, as shown on the board, whereby we are now selfing the individuals of the F1 generation. So here, parents phenotype, parents genotype. Parents genotype, we have R, W, and then we are crossing with R, W, as shown on the board. During meiosis, the genes segregate or separate, as shown on the board. This one also segregate in a similar way to form R, capital, and capital W. So the genes have segregated, and each one of them is carried in single gametes, which are haploid. So now at fertilization, what happens there? Of course, as usual, we make or draw our cross lines. This one, this allele can fuse with this one here, and also has a chance of fusing with allele W, capital. Also this one here, it has a chance to fuse with either allele of the other gene, as shown on the board. So here I will have F2 generation. F2 generation, whereby we shall have genotype RR, capital. Here, capital R, capital W, capital R. For this one, capital W. That's where we are. Then we have capital W, W. Capital W, W. What we call F2, F2 genotypes. F2 genotypes. And here we have random fertilization. Random fertilization. So now what can we deduce what can we deduce about the phenotypes of the offspring in the F2 generation? Then we can see that F2 phenotypes. F2 phenotypes, we have one red flowered, one red, then we have two pink, and then lastly one white. So you can see very well or clearly that, as we said, that the phenotypic ratio of the monohybrid inheritance in the F2 generation, which is 3:1, of course, is modified or changes to 1:2:1, and so this is due to genes or alleles being codominant. So let us meet once again